In this video, we are going to learn about digestive system of an insect in detail. So, different categories of that: alimentary canal, the salivary glands, and insect nutrition, mechanism of digestion, intestinal microorganisms. These are some different categories involved in this section. So, the digestive system is represented by a tube, the alimentary canal or gut of variable diameter and length, which extends from a mouth to an anus. It is supplemented by a pair of labial or salivary glands in many insects, gastric caecae in some insects, and the Malpighian tubules, which are exclusively the excretory organ in almost all insects. Some parasitic hymenopteran larvae seem to be exceptional, in which the alimentary canal is merely a simple tube or completely degenerated, as the nutrients are absorbed directly through general body surface. The alimentary canal is short in length in the primitive insects such as Pterygota, Epimeroptera, Odonata, Dermaptera, Phasmida, and the larvae of butterflies, moths, bees, wasps, and mosquitoes, etc. While it is enormously elongated in some insects, including Homoptera and Cycloralpha, which is nothing else but the Diptera. The alimentary canal is often longer in the phytophagous insects than in the carnivorous insects, though it cannot be strictly applied as a general rule for all groups of insects. The structure: the alimentary canal is typically divided into three main regions like foregut, midgut, and hindgut. The foregut and midgut are ectodermal structures, while the midgut is endodermal in origin. The foregut and hindgut are ectodermal invaginations and are therefore called stomoderm and proctoderm respectively. The midgut, on the other hand, represent the middle endodermal sac which connects the anterior foregut with the posterior hindgut and called the mesenteron. Okay. The foregut starts with a true oral cavity or mouth, and it is differentiated into various parts such as pharynx, esophagus, crop, and gizzard or proventriculus. All these structures differ in morphology as well as in function. The pharynx is provided with a pair of The foregut starts with a true oral cavity or mouth, and it is differentiated into various parts such as pharynx, esophagus, crop, and gizzard or proventriculus. All these structures differ in morphology as well as in function. Then the pharynx is provided with a pair of dilator muscles. It forcefully pushes the food material into the esophagus, and in sucking insects, it is modified into the sucking pump. The esophagus simply serves as a passage for food material and is therefore a seat of food conduction. The crop is ill-defined in Pterygota, Paleoptera, and parasitic insects, while well-developed in Dictyoptera and Orthoptera as a dilated sac-like structure. It is completely separated as a lateral diverticulum which opens into the esophagus by a small duct in some blood-sucking dipteran flies and Lepidoptera. It functions as a food reservoir. The proventriculus is greatly developed in Orthoptera, Dictyoptera, and Coleoptera, feeding on solid food material. So when we try to learn about the basic plan of the alimentary canal in a generalized insect, we could see a middle portion called mesenteron, stomoderm, mouth, ectoderm, endoderm, ectoderm, anus, proctoderm. Okay, mesenteron is there. Then mouth, pharynx, esophagus, crop, proventriculus, gastric cecum, then ventriculus or midgut, Malpighian tubules, ileum, colon, rectum, and anus. Okay. The alimentary canal of various insects: dragonfly, esophagus, midgut, Malpighian tubule, ileum, rectum. Cockroach, crop, gizzard, gastric ceca, then ileum, rectum. Okay. And red cotton bug, first ventricle, second ventricle, third ventricle. Lepidopteran larva, ventriculus, and pylorus rectum. And beetle, rectum in different parts. Housefly, cardia, crop, etc. The inner cuticular layer of the gizzard is differentiated into various processes like protuberances, denticles or teeth, usually arranged into distinct rows or plates. The teeth are arranged into 6 longitudinal folds in Acrididae and 6 plates in Blattioidae. Each plate is broken up into a series of transverse ridges or overlapping teeth in Tetygonidae, Grillidae, and Grillothalpidae. In the dragonfly nymphs, however, there are paired dorsal ventral plates, each bearing a group of teeth. While in the meloid beetles, the teeth are spiral, pointed, and variably developed. The fluid-sucking insects usually lack a proventriculus, except the region of cardiac valve. Besides diversity in structure and function, different regions of the foregut resemble each other in their basic histology. The wall is typically composed of the following 6 layers: the innermost cuticular lining as intima, the epithelial layer and the basement membrane, the longitudinal muscle layer, the circular muscle layer, and the outermost peritoneum. In the midgut, the gizzard opens into the midgut through the cardiac, proventricular, and stomodeal valve. It occurs as a short, straight tube in primitive insects and caterpillars, while greatly coiled and enormously long in higher insects, or modified into a pyriform or filiform sac in the mosquitoes, and even into 2 chambers, anterior cardiac and posterior ventriculus in some Cyclorrhiza, and into 4 chambers or ventricles of variable shape and size in Heteroptera. In some insects, Diaspine cockets, larvae of Apocritian Hymenoptera, some Neuroptera, and Viviparous Diptera, the midgut is a closed sac and is completely separated from the hindgut, in which all nutrients are directly discharged into the hemocoel through the walls of the midgut. It bears large number of gastric ceca throughout its length in the adult beetles. The wall of the midgut is composed of 5 layers: the innermost columnar epithelial layer, the basement membrane, and the circular muscle layer, the longitudinal muscle layer, and the outermost peritoneal membrane. Longitudinal muscle, esophagus, circular muscles, intima, crop, denticle plate, pad, cecum, stomodeal valve, midgut, circular longitudinal muscles. Structure: the crop, proventriculus, stomodeal valve complex in Periplaneta, dental area, pulvillar area, stomodeal valve, etc. See. And in case of proventricular dentition, that is the second one. Then peritrophic membrane formation, foregut, secretory cells, stomodeal valve, invagination, midgut, peritrophic membrane. The wall of the midgut is composed of 5 layers: the innermost columnar epithelial layer and the basement membrane, the circular muscle layer, the longitudinal muscle layer, and outermost peritoneal membrane. Microvilli, mitochondria, secretion, zonula continua, Golgi apparatus, nucleus, rough endoplasmic reticulum, invagination of basal plasma membrane, basement membrane. Opening of lumen of midgut, intercellular junction, cavity of goblet cell projection into cavity, mitochondrion, nucleus, infolding of basal plasma membrane, basement membrane. The internal cuticular lining is absent, and arrangement of muscle layer is just reversed than that of the foregut. The epithelial cells are of 4 types: The columnar secretory cells have some homonomous or different size, heteronomous with a brush border. The regenerative cells, either scattered or grouped into the crypts or clusters, and the goblet cell serving excretory function, and the endocrine cell secreting peptide hormones. The regenerative cells are absent in some insect strips, while columnar cells secrete holocrine, detaching, merocrine droplets or apocrine, granular secretions. These secretions represent a mixture of the digestive enzymes. Regenerative cells are there. The regenerative cells are absent in some cells, while columnar cells secrete holocrine or detaching merocrine droplets and apocrine granular secretions. These secretions represent a mixture of the digestive enzymes. First, columnar cells, they are characterized by apical microvilla border coated with mucopolysaccharide glycocalyx and about 20 to 200 nm thickness. The lateral walls are straight, unfolded, but the adjacent cells are connected to each other by separate junctions of desmosomes. The basal wall is folded along with invaginations of the basement membrane. The basement membrane may occur in lattice-like form and multi-layered, ranging from 0.13 nm to 1.2 nm in thickness. Microvilli contain fine filaments about 6 nm in diameter. Columnar cells, we mentioned. The basement membrane may occur in lattice-like form and multi-layered, ranging from 0.13 nm to 1.2 nm in thickness. Microvilli contain fine filaments about 6 nm in endoplasmic reticulum. Rest of the cell body is occupied by mitochondria. Abundant Golgi bodies, membranous vesicles, and granular endoplasmic reticulum are concentrated around the nucleus. Secretory granules are present in variable density in the cytoplasm, representing enzymes or their precursor proteins. The cells are replaced after every day after 4 days in the Tenebrio molita. That's an insect. Then goblet cells. They are scattered among the columnar cells in the Lepidopteran larvae. They are characterized by invaginated luminal plasma membrane forming a flask-shaped large cavity. Cells contain little cytoplasm and a basally situated nucleus. The basal plasma membrane is invaginated into a number of short folds. The invaginations of the flask externally enclose the mitochondria, while those of neck are devoid of mitochondria. The flask opens into the midgut lumen. They are concerned with excretion of potassium, Lycoptera, and Streptocera. Endocrine cells are intercalated. Columnar and regenerative cells, they are bowl-shaped, pyramidal or bottle-shaped, fixed basally or basal lamina and extended towards the lumen. The apical end of endocrine cells is closed by a tuft of short microvilli. The basal lamina and the basement membrane as well as apical membranes are unfolded. They are characterized by the presence of granules in cytoplasm along with Golgi body, rough endoplasmic reticulum, and few mitochondria. The endocrine cells are intercalated among the columnar regenerative cells. They are bowl-shaped, pyramidal or bottle-shaped, fixed basally on basal lamina and extended towards the lamina. The apical end of endocrine cells is closed by a tuft of 4 short microvilli. The basal lamina and basement membrane as well as apical membrane are unfolded. They are characterized by the presence of columnar cells we mentioned, then goblet cells. They are scattered among the columnar cells in the Lepidopteran larvae. They are characterized by invaginated luminal plasma membrane forming a flask-shaped large cavity. Cells contain little cytoplasm and a basally situated nucleus. The basal plasma membrane is invaginated into a number of short folds. Invaginations of a flask externally enclose the mitochondria, while those of neck are devoid of mitochondria. The flask opens into the midgut lumen. They are concerned with excretion of potassium. They are also reported in Ephemeroptera, Plecoptera, and Trichoptera. In endocrine cells, the endocrine cells are intercalated among the columnar and regenerative cells. They are bowl-shaped, pyramidal, or bottle-shaped, fixed basally on a basal lamina and extended towards the lumen. The apical end of the endocrine cells is closed by a tuft of short microvilli. Okay. The apical membranes are unfolded. They are characterized by the presence of granules in cytoplasm along with Golgi body, the endoplasmic reticulum, and few mitochondria. The granules are about 0.1 nm in diameter and thus representing peptide granules. Then next comes the peritrophic membrane, which is a chitinous envelope secreted by the anterior or entire epithelial layer of the midgut, which envelops the food container and protects genetic epithelial cells from abrasion by hard food granules and no mucus secreted in the insects. In the fluid feeding insects, it is wanting. It is permeable to the digestive juices and products of digestion and thus function as an ultrafilter. On the basis of structure and mode of formation, the peritrophic membrane can be classified into two types: lamellated peritrophic membrane and non-lamellated. Dragonfly, Leishmania secrete two peritrophic membranes per day, even during the period of starvation. The lamellated peritrophic membranes have been reported from Ephemeroptera, Odonata, Acredids, Phasmida, Lepidoptera, Hemiptera, and Coleoptera, Arabia, and non-lamellated peritrophic membrane, which is an undifferentiated single layer of uniform diameter. Non-lamellated peritrophic membrane. It is an undifferentiated single layer of uniform diameter. It is secreted by the epithelial cells of the midgut, situated in the invagination fold of the anterior cardiac valve. The opposing walls of the cardiac valve press against each other for the molding of the peritrophic membrane as a membranous tube resulting from the solidification of the molded secretion. The membrane is uniformly drawn backward through the peristaltic movements of the gut. It is found in Dermaptera, Lepidoptera, and secreted in Diptera at the range of 6 mm per hour. It is made up of superimposed layers of fine fibrilla strands oriented at 60 degrees to one another, and thus forming a fibrilla network having hexagonal strings of a force of about 4 to 10 mm in diameter and permit transport of energy but still dash complex impact. It is chemically cyclic protein and proteoglycan on glycogenation product. The proteins and proteoglycans form a which interlocks the chitin fibrils and therefore determines permeability properties of a peritrophic membrane. Proteoglycans of peritrophic matrix are acid mucosaccharides like uronic acid and form gel-like meshwork. The proteins are glycosylated with oligosaccharides. Then filter chambers. Due to the fusion of anterior hindgut with the anterior or post-embryomost part of midgut or end of foregut, a so-called filter chamber is formed in many Homoptera. The modification facilitates quick removal of excess of water from the lumen of foregut or anterior region of midgut to the hindgut, so that it allows the dilution of concentration of enzymes involved in digestion in one middle region of midgut, and also in turn avoids dilution of the hemolymph. The anterior midgut that modifies into a bladder. The water passes directly from the anterior midgut to the hindgut along the osmotic gradient, and the posterior midgut receives concentrated fluid suitable for enzyme activity. An organization of filter chamber along different facilities, families of Homoptera undergoes complex modifications. Membranicidae. Midgut wall makes intimate contact with the foregut epithelial layer after penetrating the muscle layer of the foregut. Then Physalidae. Anterior part of midgut, hindgut makes close contact with the hind part of the esophagus by coiling tightly around one another. Then Cercopidae, formation of a filter chamber occurs due to fusion of diluted anterior part of midgut with hind part of the same, and at the site of fusion and hind part of the midgut gives large number of infoldings which merge in the wall of the diluted anterior part. Then Cicadoidae, anterior segment of the midgut is fused with the anterior end of the hindgut along with the Malpighian tubule. Both the walls are fused compactly and bound with own basement membrane. The filter chamber is formed within the anterior midgut. Coccidae, both the ends of the midgut are fused with one another, and this fused complex is enclosed in the hindgut. In Pentatomorpha, the midgut is divided into 4 regions, and the ceca of the fourth region actively remove water from the hemolymph, avoiding dilution of hemolymph, and conduct backward flow of water to the rectum. The hindgut. The midgut opens into the hindgut through the pyloric valve. At the junction of midgut and hindgut are developed the Malpighian tubules. It is differentiated into 2 regions, the ileum and the rectum. The ileum is sometimes differentiated posteriorly into the colon. Orthoptera or iliac sac, dragonfly means. The rectum is an enlarged sac, and the epithelium is alternatively differentiated into thick and thin regions, resulting into the formation of 6 rectal pads of the tall columnar epithelial cells in many terrestrial insects. They absorb water from the fecal matter. The epithelial pads are replaced by 4 large papillae in Diptera and Hymenoptera, 6 rectal gills in dragonfly nymphs, and the anal sac in Tricinida. In histology, the hindgut resembles foregut with some modifications, such as the cuticular lining is very thin. Longitudinal muscles form outer strands which run parallel to the rectal pads, and the cuticular muscles are weakly developed, and the epithelium forms thick excretions of rectal pads, usually 6 in number as described earlier. In adult Odonata and Orthoptera, each pad is composed of a single layer of cells, but in Neuroptera, Lepidoptera and Hymenoptera, it is made up of 2 layers. In Odonata, they function as respiratory and osmoregulatory organs. The nerve innervation of the gut. The foregut is innervated by the stromoidal sympathetic nerve system comprising the frontal, hypocerebral and influvial ganglia. They are nerves and the recurrent nerves. The hindgut receives the nerves from the last abdominal ganglion. The midgut may partially be innervated from influvial nerves anteriorly and from the nerve last abdominal ganglion posteriorly. While in the larvae of Oryctys, the entire midgut is innervated from the stromoidal nerve system. The nerves are mostly motor nerves and are concerned with the control of muscles of the gut. Presence of sense organs, receptors in the pharynx of Pediculaneta has been reported recently. Then trachation. The gut is richly supplied with segmental trachea. The trachation, besides supplying oxygen, provides mechanical support and fixed position to the gut in the body. The tracheoles penetrates the epithelial cells, providing enough aeration to the midgut cells. The tracheal innervation differs from one part of the gut to another. It is highly concentrated at the crop, midgut and rectum than other parts of the gut. And then the salivary gland, a pair of lobulated glands lying in the thoracic region and associated with the labium, are commonly called the salivary glands. The glands, wait a minute, so I have completed the alimentary canal. Okay. The next is salivary glands. A pair of lobulated glands lying in the thoracic region are associated with the labium, are commonly called the salivary glands. The glands open anteriorly into their respective lateral salivary ducts, which unite and form a single median salivary duct in the cephalic region and open into a preoral cavity between the labium and the hypothalamus. In a large number of insects, they secrete saliva consisting of enzymes, mostly amylase, invertase, and sometimes even protease, lipase, but never cellulase. In the larvae of Lepidoptera and Hymenoptera, the salivary glands secrete fluid, while in blood-sucking insects and anticoagulant, which mainly in state of ingested process. In structure as well as function, they differ among insects, mostly in relation to the nature of food. Hypopadding, common duct of gland, efferent salivary gland, common duct of reservoirs in gland, salivary gland. Posterior lobe, lateral lobe, anterior lobe, salivary duct, accessory lobe. There are two pairs of salivary or labial glands in the honeybee. One pair is situated in the ventral region of the head, called the cephalic salivary gland, and the second pair is situated in the thoracic region, called the thoracic salivary gland. The cephalic glands are actinophyte and secrete the lipoidal and mucoproteinaceous secretions which are used in wax production and for moistening the food material for chewing. Thoracic glands are tubular type and secrete the proteinaceous secretion consisting of invertase and other digestive enzymes. Besides salivary glands, there are paired mandibular glands and two pairs of paramount and royal jelly producing pharyngeal glands in the worker structure. The salivary glands contain large number of lobules or acini and therefore retain great sponge-like aciniums from the Orthoptera, Dictyostera, Hymenoptera, and large number of insect studies, Lepidoptera, Physcomoptera, and Diptera, as well as the thoracic salivary glands in Hymenoptera. They occur in a tubular form. Each salivary gland is provided as a reservoir in some insects, Odonata, Dictyostera. Each acinous gland contains two types of epithelial cells. The large secretory cells at the center and small parietal non-secretory cells at the center. The glands are covered externally by a thin transparent peritoneal sheath. Each acinous gland opens into its own duct, which transports saliva first to the lateral salivary duct, and later on both the lateral saliva ducts open into a common salivary duct. Saliva is finally discharged in a cavity or pouch-like region, salivarium of the hypothalamus. The secretory cells rest on a distinct basement membrane, and the cytoplasm contains the granular endoplasmic reticulum, Golgi mitochondria, lysosome, and membranous vesicles, active in large number of proteins, primary granules, associated digestive enzymes. The parietal cells contain extensively folded microvilli enclosing mitochondria and form the duct. The parietal cells may transport water and sodium and potassium ions in the urine of the duct through a mechanism of acetogenesis. The acinous glands are innervated by the nerve running from the subesophageal and the stomodial sympathetic ganglia, mostly releasing dopamine, serotonin, and octapamine. It is usually noticed that dopamine stimulates secretion of the fluid, while serotonin induces synthesis of salivary production enzymes in the secretory cells. Saliva function. The saliva contains the carbohydrate like amylase and invertase predominantly and lipase in a little quantity in most of the phytophagic and plants are feeding insects. Saliva of carnivorous insects, in the contrary, mainly contains the enzyme protease. In the insects which feed on selective kinds of food, the saliva contains specific enzymes such as chitinase and cellulase, macroterminase, collagenase, riboflavin, hyaluronidase, pectinesterase, galacturonidase, oxidase, antipyretics, and catechol oxidase, peroxidase, milkweed, bug, and of digestion of fungus, root cellulose, vertebrate tissue, insect, cuticle, and plants are collected. Saliva in the honeybee contains predominantly the enzyme invertase that catalyzes the conversion of nectar into honey in the workers only, and it remains absent from that of the queen and drone. In the worker honeybee saliva, various enzymes are present, such as acid phosphatase, naphthol KSBI by phosphohydrolase, catalyzing protein synthesis, leucine arylamidase performing hydrolysis of alpha glucosidase catalyze the conversion of nectar into honey, and beta galactosidase in young bees involved in energy supply. In blood-sucking insects, however, the ideal digestive enzymes are largely from saliva, but on the other hand, the saliva contains homeostatic substances like platelet aggregation inhibitors, enzyme upgrade, anticoagulant, antithrombin, and vasodilators. Plasticae like substance. In some Lepidopteran and Phycosteran insects, the salivary glands are the large, with the protein fibrose conservation and producing silk to construct protoplasm. In the last steam star, silk moth larvae or silkworms, the fibrone is converted into a thread after passing through a silk cut, while sericin is converted into a gum-like substance that cementing firmly the silk threads together.